This is a production of Cornell University.
Season is about to start, hopefully in about 20 days. So would you yeah. So the video that's playing right now is was taken a few days ago, and it's one of our orchards where we measure lots of things, productivity and yield efficiency and so on and so forth. I'll be talking about all those some of those verbiage later. But it's one of the ways that we're evolving. Given the title of my talk, things started 50 years ago. I'm 51 years old. And so you can imagine how long it's taken to get here.
So you started the program when you were
I was 1 year old. I was just planting those rootstocks right there. It's easier to reach the bud, the graft union when you're tiny and you're little. But no, it's nice to be standing on the shoulders of really great people that started the breeding program a long time ago. I'll have a few pictures from the 1970s and 1980s. I took a few hours this past week and got into Herb Alwinkle, who is this fellow right here, and this fellow right here, who had a collection of the old slides that used the slide projector. So I scanned a few of them, and you'll see some of them. But the breeding program was essentially the idea of Jim Cummings. Here he is last year, and here receiving a very nice award from a group. I think it was ISHS, and then the breeding program was joined by Terrence Robinson, and of course Herb retired a while back, and so we've now made a formal collaboration with Dr. Abais Khan's. But there are lots of components to breeding programs and other people working on it, and so I'll be highlighting some other people that have worked on the breeding program, like Charlie, who passed away a few years back, and Todd Holler and Sarah Bauer. They're both of the technicians that work in the breeding program currently and do a great job. So here's an old picture that I took from Herb's slides, and here's Herb, and he's presenting a poster. But I think that's also Herb in England, and featuring a beard of some sort. But a lot of people have contributed and continue to contribute. A lot of students have participated in the past few years and in the breeding program, doing all sorts of things. So we have Abby, who is more of a stable technician. She's the propagation specialist in our program. And some went on to become Cornell students. Here's David, and then we have some more that have actually joined us going sailing. That's one of our sailing parties. And we had students coming from Hobart William Smith and Utah State that participated in the Summer Scholars projects. Also students from BYU, and here's one of our meetings where we're discussing what to do in the breeding program, all the building projects and things that we were undertaking this past year. We have more students, more people that have worked on the program. I don't know if you're familiar with some of them. I think he was Jim Giovanni's technician and then earned his PhD here. And now he's Dr. Quinn. So he started back in 2002, 2003 with us. We have Jack and Jane O'Reilly, Yichen Wan, and my neighbor, Ava. And then Chris Daffler, I think he's a medical doctor now, and Darius Kiklis, who was a postdoc or visiting scientist in the Terence Robinson program, did a lot of work on roots and replant resistance and things like that. More people. There's Phil Forsling, and Phil was one of the initial technicians that worked in the breeding program 50 years ago or 40 something years ago. And Phil went on to become retired as the research leader of the USDA unit. And how appropriate in this picture that I found among my things, he's standing next to a fire blight stricken tree, and there's fire in the background. So, what a nice picture. Stefano Musacchi from Italy, who was a visiting scientist at our lab. That's Herb Gustafson, and then Boric, Hela's husband, helping with inoculations. We have Kathy, that is, you'll see another picture later, measuring things like fruit numbers and suckers and whatnot. Another picture of Herb with George Sundin, who is in Michigan State. So a lot of people, it's amazing how people it has taken to get to this place. And I have to recognize all of them. A few more students, postdocs are good, but students are great because of really the very nice contribution that they make to the program. Here's Ali. I think he's present in this room. Raise your hand, Ali. There you go. There's another picture of you menacing with an X-ray machine in the next few minutes. And other students, that's Nicole Ojudice, who worked a lot on evaluating our plant material for fire blight resistance. And then two other students that worked on, and they're part of the Adams family, and they own a nursery. One of them is the director now of propagation of Willow Drive Nursery, and the other one is Scion Varieties. So they've all gone to do great things, but in the process, they've all studied apple rootstocks. Okay, moving on. And of course, I have to recognize all my colleagues and collaborators that are, and this is just a small list of US-based collaborators, but we have obviously over 100 people that have collaborated with the breeding program, and just in the US, and maybe if we go into other countries, we also have that probably the same number. So I recognize all of them. I realized that evaluating apple rootstocks or working with root systems and rootstocks in general is a very difficult thing to do, and I appreciate the work that they've done to collaborate. So a little bit of a review, and real quick, what does a rootstock do in the orchard? And we've learned a lot in these past few years about how a root can modify the architecture of the grafted scion. But and on top of doing the architecture, you'll see later on that it does a lot more than that. But and other things that it does, obviously, it interfaces inside the soil with microbiota and soil particles, collects the water and all those good things to make the scion more productive. And it has to be resistant. Rootstock is not, rootstocks don't live all by themselves. They're not isolated from the rest of the orchard. In fact, they are an integrated part of the orchard system to make that orchard system productive. And so all those interactions, if you can imagine, you somewhat have to breed for or account for when you evaluate a rootstock. You can't just focus on soil pH or disease, when a rootstock goes out, you kind of have to have the whole spectrum of those interactions covered, or as many as possible. And as you'll see later, sometimes you know, the work that we do as plant breeders is where we can't come up with perfection. Sometimes we can, maybe, but or we can approximate. But there will always be those interactions. If you can imagine those interactions happening in microclimates around the world, which is what we currently are testing in the Geneva breeding program. We have experiments from South Africa to South Korea, to New Zealand, Japan, and all kinds of other places. So, so if you can imagine all those interactions, they are obviously, it's difficult to account for all those different microclimates. So in the past few years, we learned about some very unique root technologies that the Geneva Rootstocks have imparted to the science, like increasing productivity that's worth a lot of money, the branch angle modification. Basically, our rootstocks are able to flatten the branches of trees that are grafted on top. So and then replant disease tolerance. This is the communication in and the survival in the of the root systems. Woolly leaf resistance, and they're all worth a certain amount of money for apple growers. Some apple growers, you know, want me to work on mice and voles and vole resistance, and I said, you know, it's going to take $250,000 and a lab to raise voles, and so they said, no, okay, that's too much. But we can put that list. We can go down that list quite a bit, or I add to that list to see how many things. So a little bit of history, 50 years. Okay, 5 minutes. Let's do 50 years in about 2 minutes. This is when things sort of started right here in 1968, more like 1969. Robusta 5 is identified as a good parent. I have some pictures later on. And it took this long in order for the first release to come out, G65, and then the second release was G30. And G30 is still being planted today. It has you know, some difficulties. And in 1995, Dr. Cummins retires. The material was viewed by apple growers as very valuable, but the PI, or you know, the researcher, was not replaced so the growers went to Congress and they said, okay, so we need that material to be preserved and to be advanced into a finished stage. And so the USDA sort of stepped in through an act of Congress, and they set aside, you know, back in the days when they had earmarks, they set aside a certain amount of money, and they hired Bill Johnson, who was my direct predecessor, and he was there for about 18 months, and then left and went to breed, I think, eggplants or something like that. And then that's when sort of I stepped in, and we took the lead of the breeding program. That happened in 1998. And back in 1998, maybe the production of Geneva rootstocks was less than maybe 50,000 pieces a year. And we've released, we continue to release several other rootstocks. Dr. Alwinkel retired right here. And right now we're just about 10 million trees being planted every year, if not more. So 10 million is conservative, I think. And the demand is still much higher than the production. I remember walking into a meeting with Washington growers when I first started, and they said, "Where's the plant material? What happened to it? You know, we want it. We want to test it. We want to planted, and we didn't have enough production capacity. And that was perhaps one of the hurdles that we have overcome through research and also good technology transfer. I have a couple of slides later about that. Okay, so I'm going to go through really quickly with sort of the fact sheet on many of the rootstocks that we released, just so that you know a little bit about them and about the qualities that they have. I think we have about 3 million rootstocks that are planted every year of G11. It's resistant to fire blight. Okay, so all of these rootstocks were bred to be resistant to fire blight. High yield efficiency, which means that the rootstock helps the apple tree produce many more apples per tree size. So you can essentially say you can produce more apples per acre. But obviously it's not perfect. It's susceptible to woolly apple aphids. Then we have G202, and this is our workhorse. It's been around since the 2000s. It's a really good rootstock. About half of the market share in New Zealand is planted, trees. Half of the trees are planted on this rootstock in New Zealand. And in the US, we have probably a production of about 1 million or so. G202. Here's, I told you that I scanned some pictures from old times. So this is Charlie standing next to a tree, and you'll see in a few minutes that we're still doing something similar, but hopefully with drones we're able to do something a little bit quicker and better. But this is G202 back in the 90s. And this is G935. This is an experiment in Italy that that we have with G935. It's really gorgeous, very productive tree, and we have about 2 million and a half to 2 million trees being planted every year on this rootstock. It really has some potential, but lately we've found some problems with some viruses, and so we're concerned about. We're concerned about that. We're finding out what it is that obviously wasn't tested before we released it and bred for it. But it's also a very good rootstock. G210. We released it for the apple processing industry because some folks wanted a little bit stronger apple tree, a bit more vigorous. And so I think, you all know who Ian Merwin is, right? I think he's got an orchard with G210 and a lot of his cider varieties, and it's showing out to be a good, decent rootstock for that. 890. This is the most vigorous of our releases, and if you recall, yield efficiency, this rootstock, even though it makes a big tree, it produces with a yield efficiency that's similar to M9, which is a very dwarfing rootstock. So it's very productive. And we've tested it with many different varieties and many different things, and it's just an amazing rootstock. G214 is another one which also is being propagated. We're having some issues with the micro-propagation. Anybody that has some ideas on how to root G214 can probably make a lot of money, because that is the I guess the block right now for high implementation of this rootstock. And A14, another rootstock that is was released based on a few things. So it's a good propagator. You see, this is a propagation bed where with A14, and it has a very nice root system. But one of the things that was very nice about it is this feature right here. It promotes larger fruit size given comparable crop load. And that's, you know, for an apple grower, a slightly larger apple, at least in the US, means money. And so if you're able to have a rootstock that consistently produces larger, slightly larger apples, you can make, you can take that to the bank quite a bit. And we've shown there's actually good economic data about that. 969, this is another one of our recent stars that is taking off as a rootstock. It's here you see it in Italy, and that is again Charlie with 969 in the background. And that's Nicola Della Betta, my collaborator in Italy, who has planted these trees. And what's unique about these trees is they have 3 buds that have grown independently into a structure, you know, into a flat structure with multiple branches. And this is becoming something that's sort of desirable in some places in Europe. Okay. Moving on, and this is our latest release. I'm going to move on to, I'm going to pass it on, but one of the special things about this rootstock, it's dwarfing and everything, but one of the special things that we found out through some experiments is that the rootstock actually promotes higher flowering in the top third of the apple tree when chilling requirement is not being met. So it reduces the chiller requirement of the Scion variety. We've done a lot of work looking at this rootstock here in the U.S. and we found out that it, you know, the same that true it was in Brazil, it's also the same and here. What we're trying to find out is QTLs and the genes behind the this reduction transmissible to the Scion for that. There are a lot of online resources for Geneva rootstocks. If you want to go online and look it up, you're more than welcome to look more information up. Okay, so why did it take 50 years to get here? And so this is sort of the my breeding chart, or the things that we do in order to come up with a rootstock. And in a few minutes, I'll take you through the life of an apple rootstock from conception to commercialization. You can see right here, you have years 27 to 30. It's a little bit shortened if you use molecular markers, which we do use in our breeding program. But it just takes a long time for a tree to grow into full maturity, and that's the name of it. And then anytime you test it into a different locality, you're going to encounter different things: cold, dry, you know, whatever. And so you don't know how it performs in a certain place until you actually test it. So I'm going to take you in a journey through the development of one of our star apple rootstocks. We probably sell about 4 million pieces every year, and should be probably 7 to 8 million of these in the future. And then I'll try to focus a little bit on what's new in apple rootstock research. So G41, it's a star. And really productive rootstock. It's resistant to all kinds of pests and things. But it really, and I think the cross started, and I'll show you, before we got to this point where we know about G41 so well and about what it can do in many different environments, it took a while. One of the latest varieties, I don't know if you've heard about Cosmic Crisp, but very susceptible to fire blight in Washington State. Probably see it in a supermarket this coming year. But these were this were probably about 300,000 apple trees of Cosmic Crisp grafted on G41, and the bud take was very, very nice. So, breeding program. Before you start a breeding program, you have to have sort of an idea of what you want to do. And back in the day when I was trying to choose whether I would go to Wisconsin-Madison or North Carolina State or other universities, I didn't know who Randy Gardner was, but now I do very well, because Randy Gardner was the student that did the initial work that recognized some of the resistant material. So you have to start from germplasm and testing germplasm before you make your crosses, and you have to learn about your germplasm. So here you have the first table, one of the tables in his thesis, and talking about the seedling inoculations. And here's Malazex robusta 5. He mentions it directly. So you know, think about that. That's the starting point where you have your objectives and you think about what the future is going to be. Parents were crossed. Here we were using bees to do that. Harvest the seeds, you know, that's okay, fine. So that took time, and it takes a lot of effort every year to do that. But then the unique things that happened was this disease screening methods that Herb and Jim Cummings and Gustafson put together. And this is what, you know, the intention was to kill as many rootstocks as possible, plantless as possible. Very successful at that. We're trying to restart that. But these are old pictures that I took, actually pictures that I scanned a couple days ago, and much the same of what we were doing now, or we did back then. So we don't mess what was successful. And that's Gus. Then we also do a seeding inoculation for fire blight. We don't need molecular markers now for this, because yeah, if it shows anything, just throw it away. We don't need anything that is not resistant. This is again a picture probably taken in the 1970s of the inoculation that they were doing. And then we have to take that plant that survives all of that, and we plant it. Well, right now we don't plant them directly. Right now they go through a series of tests with molecular markers for our marker-assisted breeding selection protocol. And I'm not going to have too many, or actually I have only one picture of some molecular markers. And I did that because I could go on forever, and I don't have forever. If I start talking about you know, gene expression and all of that other good stuff. It happens. But the important thing is that the plant makes it out into the field. We start to propagate it. We get a initial product, and this is the product that gets sold to the nurseries that make apple trees. And so we get a first look of that product. We evaluate the rooting. Then we take that and we make trees, because that's what we evaluate. And so these are older pictures, and then we have Alexis right there that was doing that a couple of days ago. She was bench grafting trees so that they can be planted and evaluated. And we plant all sorts of orchards, and those orchards get evaluated for 8 to 12 years. We kind of know what the rootstock is going to do at year 7, and then when we find out what's working in that orchard, we go into a stage where we take that material and we want to know more about it. So you know, how do you find out more about a genotype? Well, replicated experiments. But how do you run replicated experiments with 4 or 5 plants? You can't, and you can't do, you know, you have to, just like you do in corn, you have to bulk up the material either by selfing or, you know, hybrids or whatever. And so that's largely the role that Abby has, and she has many different ways in which she does that. And that's great, because now we've shared our material with many other different collaborators in all over the world and all over the US to find out what these rootstocks do. So we continue that and continue evaluating the material. We run some of the, so G41 had to be run again to test for fireblight resistant, but now instead of using a mixture, we do perhaps separate strains of fireblight to hit them. We'll have aphids, test again. And then again, new orchard trials. But these now become the elite orchard trials where we have the material that perform well in the first set. And so what we're seeing now is basically, hopefully, good stuff. Well, thanks to molecular markers, we now can go a lot faster here. And we can select things. But this is the way we kind of used to take notes and everything, and then you had to transcribe and you know, and you had errors and so on and so forth. Well, a few years ago, some folks in the Midwest started working on this app, and this is like the most wonderful. How many of you have heard about of Fieldbook? 1, 2, 3. Okay. So if you run any experiments, any replicated experiment, get the app. Okay, because you can enter your experimental design and then collecting data, images. Here I'm measuring suckering. All of that stuff is just like very, very, very easy. So I would recommend you do that, and that's something that we used to do by hand. And it shortens time to come to conclusions and evaluations. Okay, 10 more minutes, so I have to go very quick. So this is also something else that we used to do to the advanced orchards. We used to spray them with fireblight in order to find out whether they're truly resistant. Here's G41 in one of those orchards, planted many years ago, and here's Charlie again next to it, and you can see it's a dwarf, productive, flat branching, nice tree, which was very impressive. So we used to do that. We still do it sometimes with the background, but then we are trying to graduate into the this millennium by using drones. And so Martin Hooker is our drone specialist. He's actually licensed to do that. And so there's a drone that's taking pictures of all the trees, and we're able to look at those structures and things and do measurements afterwards, so that we don't have to get blown in the wind like it happened right here. All right. Okay. So we go through a commercial propagation trials afterwards, where we share the material with nurseries. And so I know all of our nurseries in the US and many of the nurseries internationally face to face. I visited them. I know their growing conditions. Terrence also has done that, or Dr. Robinson. And that's important because that relationship allows us to understand how things are working at their locations. And it makes us realize how important they are for the apple industry. They're the conduit by which our product, Rootstocks, gets transferred to apple growers. And so we have to, we can't just release material and just put the blinders on and say, it's going to do something. And we actually actively engaged with several of our nurseries. We have a great technology transfer specialist here at Cornell, Jessica Liga, that helped us out. And I'll have a few. But what we do at that stage is when we have bulked up the material, we collaborate with a group of scientists that's been meeting for about past 35 years, evaluating rootstocks in a replicated manner. So these are experiments that have the same genotypes, the same number of replications, and they're planted all over the US. And they've received, the group has received commendations and awards because of the type of work that they did and the impact that it's had. Literally millions of dollars, if not hundreds of millions of dollars. And the other point that I wanted to make is that by doing this replicated trials, we create a lot more knowledge, because when you add the information that you have for one rootstock onto all these different trials, you find out that by the time that you release it, you have about 300 to 400 trial years at different locations, and that's valuable. A little bit of something. So we've had in the breeding program, we've had to make choices. So do we choose grower industry over fancy science? And hopefully we've always chosen for the benefit of growers. I find sometimes that as scientists, we can get sucked into it's called a rabbit hole. Is that correct? Of RNA-seq or this gene or whatever, and whereas our main role is to produce apple rootstocks for apple growers. Not that fancy. I mean, the reviewers don't like to know that if M9 is less productive than G41 or whatever. They go, who cares? Or most reviewers in the fancy science, but growers do. And so sometimes the interest of reviewers, journals, granting agencies as well, and all kinds of things are not very well aligned with the interests of apple growers. And so I have a big folder of unpublished science that if I retired and then spent the next 10 years, I could probably cover everything. But afterwards, G41 had to be tested on farms. So we also have this relationship with farmers. And here's Bob Black in Maryland. If you go on 15, Catoctin Mountain Orchards, they have a field trial. And I love this guy. But I also love all the other apple growers that I interact with. And they get to experience the rootstock, the plant material, and get to give me feedback. Oh, that tree is too big over here, or it didn't produce any apples, or it went biennial, or you always hear something. And then we get to see it also in different locations. The field of apple rootstocks in Washington State. The reason it's so dark is because that was during the fires that they were occurring 2 years ago. But that's G41, and it's being budded or grafted. We also, these field trials with growers help us realize how good these rootstocks are and send that message to the apple growers. So this was what the orchard looked like initially. This is in Utah, in Spanish Fork, and it's a fire blight area. Like it's like one of the best areas to test fire blight. And this is the orchard afterwards. I think it took only 2 years for all the M26 trees. We had planted our trees interspersed into the rows, and so our trees survived. Now, and so they had to take out the orchard. They did. Fortunately, they took all of our trees and transplanted them to a different location. So we still have a field trial, but now it's only Geneva rootstocks. Now, last year, and I want to continue mentioning this, last year they had a major fire blight epidemic in Washington State. I don't think we lost one Geneva tree, but the folks probably there lost about 100 million worth of trees. So I'm going to go quick. More trials and more field trials, and so and this is perhaps something that I need to mention. we use molecular markers in the breeding program, and they're worth some money in terms of selection. They save money because we don't have to plant so many trees in the field or don't have to evaluate so much. But perhaps the most impactful use of molecular markers that we did, knowing what the genotype was to find out what was going on in the industry and the mixtures and things that happened, making sure that things were correctly. So we caught situations where 100,000 plants had to be thrown out. Okay, so 100,000, that's, you know, if you do $5 a piece or whatever, you do the numbers, okay? And that was maybe a $50, $100 reaction to do. So sometimes, you know, we focus on science, but things actually, molecular markers actually have a very nice thing. Stage 10, micro-propagation, and then we release it. And so I was talking about many different nurseries, and we do work with a lot of these nurseries. But research continues after release, and it's amazing how much more knowledge we had to build up about G41 and other rootstocks after it was released, after it was made public, after I wrote the patent. Now, the cross was made in, I think, in 1975 or 1976. So, and it was patented in 2006. But these are replant experiments that happened and where the results were out in 2008, 2012, and so on and so forth, that show all the good things that G41 has beyond what was initially selected for. And so here's woolly apolephid experiments that where we found out that orchards that plant this rootstock don't have to spray anymore for woolly apolephids because the woolly apolephid dies in the root system. And so this is an experiment in Italy where 2 different rootstocks were planted, and one was covered with woolly apolephid, actually this one, and the other one was completely clean. So, and then, you know, obviously propagation experiments that took place and more propagation. But then we have those situations where I talked about, and I have 2 more minutes. I know you give me a time, but where things sometimes don't go well with a particular rootstock and in a particular sign combination. And these were actually about 50,000 trees worth about $10 a piece, maybe less, I don't know. But they were gorgeous trees, and they were huge, and because they were so huge and nice in the nursery, they all got blown in the wind and they broke off right here because G41 happens to have some sort of a graft union strength problem. We call it a quirk. And it doesn't have it with all the sign varieties. And so we had to do research long after it was released, to study what worked. And, you know, we broke all kinds of trees and with students and looked at the application of phytohormones and did molecular work on this and all of that. And, you know, looked at the hormone through xylem extraction, found out that hormones were actually different in the different rootstocks, and all of that is just expensive works, but still, you know, after the rootstock was released. And this is okay, the cell book. This is Jaume Lordan. He was a postdoc in Terence Robinson's lab, and he's the one that did the work on hormones. But how valuable it is to understand that, even though it's, you know, after release. Another thing that we did, I have to go quick on this, was to find out what, you know, rootstocks kind of get nutrients from the soil. That's one of the things that they do. And what we found out is that they have some different qualities with regards to the ability to uptake nutrients and transmit them to the scion. And so if you see right here, blue is low, red is high. This is actually boron. We measure boron in leaves and fruit and peel and so on and so forth. And what we found out is that for some nutrients, And this is over 4 years, okay? For some nutrients, some rootstocks are very, very well dialed in genotypically to absorb only so much of that nutrient. But what happens if you make fertilizer recommendations based on these nutrients, on these rootstocks? You end up, you know, over-prescribing too much medicine, especially when you plant these new set of rootstocks, which are a lot more efficient at that. So we're dealing with finding out all of these new things about these rootstocks many, many years after that. You know, obviously the nutrients affect calcium and bitter pit, and so here, perhaps this is going to be my, well, this is a new method. So 50 years ago, 40 years ago, 20 years ago, even a year ago, we used to send in material to a lab to measure potassium and calcium. Well, we have this little instrument right here that allows us to measure potassium and calcium. This is Sarah Bauer, our technician, and this is an R of 87, and the R square is for bitter pit prediction. So it predicts bitter pit, and it's amazing at doing that, and it's an amazing piece of, you know, an amazing instrument. So new things I'm going to go through here, and this is some of the research that we're doing with aeroponics, looking at root systems. This is a couple of weeks ago, and now we have, I think it was about 100,000 plants right there of G41. And here we're getting more G41. It was getting budded. So a lot of meetings, a lot of people talking about it with the trees in the background, and a lot of scientists engaged. So it doesn't take, these are folks from Kazakhstan that are interested in planting some of our, and testing some of the rootstocks. But good tech transfer doesn't happen in a vacuum. You need to do it. You need to work on it. And so we were fortunate to win an award against labs like the Sandia Labs, NASA, the Naval Research Laboratory for technology transfer because of the work that we did with our customers, apple growers, apple nurseries. And so a lot of that work happened with Cornell. Here's Jessica Liga, and visiting our nurseries. We want to be fair to growers and give good opportunities to our growers. And so we did a lot of work on that. Crowning jewel, I guess, for G41 apple rootstock. It was named as the from outstanding fruit variety from the ASHS. So all of those things take a lot of effort and a lot of people. And I'm really grateful, really grateful for all of those that participated in the 50 years. I'm sure that I missed some people that I didn't recognize, and I'm sorry about that. But it kind of gives you an idea of all of the work that goes into developing a new root system or a rootstock. It's a lot more work than cucumbers. I'll tell you that. So with that, I'd like to thank you. And
well, we are running a little over time. We have time for one or two questions, either from Ithaca or Geneva. Gennaro, I'll ask one.
Yep.
How do the introductions from your program, whether it's G41 or others, actually make it to the growers?
A couple of ways. So they usually, if it's a test agreement, if they're testing it, we develop the plant material and we actually give it to them to test under a material test agreement. But if they make it through the nursery. So nurseries are an essential component of propagating our material, and our nurseries are licensed to propagate 41 and things like that. But for the testing, we kind of go directly to them. and we pick and choose the best. Yep.
Does fire blight resistance in the rootstock transfer to the scion, or like why is it important to have roots that aren't susceptible to fire blight when you think most of it coming through
So,
the top?
yep. So the question is, does fire blight resistance move to the scion? So, and the answer is yes, maybe. So we had some experiments that showed, in fact, this was the first paper published on graft transmissibility of anything by rootstocks, which showed that the scion is modified transcriptionally. So there are transcription changes that happen because of the rootstock. So perhaps that's occurring. We have some hearsay and had some proof that scion material had less incidence of fire blight on the tops. But I think the biggest saving element compared to a susceptible rootstock is the fact that the rootstock is still alive when you have a fire blight strike. But if it's a susceptible rootstock, the rootstock dies. What happens when the root system dies? You lose the tree, so there are benefits in keeping the tree healthy or alive, where the strike that happens on top, you're able to prune it and overcome that. But there are also perhaps benefits that are graft transmissible, and that research has happened, but I think a lot more could be done, especially with a larger number of rootstocks.
Okay, we're out of time.
All right.
Thank you so much.
Thank you for having me.
This has been a production of Cornell University, on the web at cornell.edu.